Hello, everyone. I'm Emilio. I'm an oyster researcher at Pearl. First, thank you everyone for coming, and also thank you the audience for watching us through Zoom. And we are so happy to have our student Elissa Tuten and other working with the Pearl aquaculture team. So they are engaged in the whole process of oyster production and algae culture. And I believe they have become the aquaculture generalists now. And unlike other lab work, oyster aquaculture seemed less interesting and hard work. So they repeat work every day, day by day, and clean the tanks, feed the oysters, wash a lot of bottles. Yeah, but we feel very accomplished when we see the oyster from a small egg become granulates. And we also feel very sad when we see the oyster died for some unexpected reason. So they are emotional work. So I will give talk to Elissa and Kyle. They will introduce their story. First, Elissa will give a talk on behalf of the whole aquaculture team. She will talk about comparison of 3 algae styles for Eastern oyster production. Let's welcome Elissa.
I'm very nervous, guys. Okay, so my name is Elissa Tuten, and I will be producing the project of comparison of 3 algae diets for the Eastern oyster production. Isn't it pretty?
Yes.
Worked very hard on that. Thank you. So our background, the hatchery plays a very important role in our oyster aquaculture system. We work only to first the survival and the growth of oysters and producing the oyster seeds for our local growers. The microalgae is the main source of food for oysters and is a very critical element throughout the whole process of oyster production in our hatchery. Our first picture up here is the Pearl hatchery downstairs. Picture down here is our microalgae. This is the main source of food for our oyster seeds or oyster babies. So our 3 microalgae projects. The first question we asked ourselves was which algae project would positively impact not only the growth of the oysters, but the quality and quantity of their larvae. Our first algae is our lab cultured algae. This algae is a live algae and it's very fresh. It's favorable to oysters because it's like the algae they have in their water, ocean, or estuary. So it's very labor intensive to make this algae. It's also very expensive for the tools and personnel that you need to have to create someone who knows the work and how to grow the algae on their own. And it's also sometimes very easily contaminated. Our next algae is the Shellfish Diet. This algae is very low cost and common for hatcheries to use for their oysters. It's also easily stored and very dense, so using a little bit of the algae to feed a lot of oysters. But the cons is that it's a dead algae, so it's not as favorable to oysters as the live algae is. The next algae we have is a new algae. It's a Vespa. This algae is from a patented company down in Florida. It's a live algae. It's high density and it's very easy to store and use. The only con is that the cost. The cost is more expensive than the Shellfish Diet, but less expensive than our live cultured algae. Here we have the Pearl. So our objective, we are here to compare the effectiveness of the 3 algae projects for the oyster broodstock fertility, the larvae growth, and the survival. And we also want to validate the Vespa as a new company for alternative feeding for the potential for commercial style oyster aquaculture. Our first stage is the broodstock conditioning. So here we want to see which algae would produce faster growth development, whether it be the Shellfish Diet or the Vespa. Our next stage is the spawn and larvae culture. So here we want to see which one will have produced higher survival and faster growth, whether that be the lab algae, the Shellfish Diet, or the Vespa. And our third stage, again, we want to see post-metamorphosis, which one will have a higher survival and faster growth between the 3 algae. Our first stage is the Bluestack Conditioning. On April 15th, between April 15th and April 22nd, we increased the water temperature 1 degree Celsius each day until we reached 22 degrees Celsius. And on April 23rd, we started feeding with the algae between the same densities. Down below, we have the Shellfish Diet. This is like a brown color. And we had the Vespa, which is a greener color. This is our paraplastic pump. It is pumping the algae into each selective oyster container. And they have a continued flow of food. We clean the tank 3 times per week, and I have much trauma cleaning those tanks, I will say that. And we recorded the daily temperature and salinity of the water each day. Sometimes if it rained or had a heavy storm or a very hot day, we record that as well, because that can change the salinity levels. So this is the comparisons between the Bluestack condition of the oysters. We have a Vespa and we have Shellfish Diet. You can clearly see the difference of fertility between the two. The shellfish is not as plump and ready to spawn as the Vespa, and you can see the difference of the color as well. And we also compared the weight between the two. This is our Gamete Grading system. We use this system when trying to decipher which eggs are better to use for spawning. So our first grade is the less likely grade we use. It is deteriorating eggs, are not as round, and we don't see a clear nuclei in the eggs. The second grade is the more favorable grade. It has better eggs, both good and bad in them. And this sometimes if the eggs aren't good, they look like little avocados. And then the third grade is they look more rounder, they're very good, and they have a better nuclei. And we usually use grade 3 for spawning. Our graph at the bottom is showing the difference of grading systems between a Vespa and Shellfish Diet. You can clearly see that the Vespa has a higher average of grading than the Shellfish Diet does. So this is our relative egg number. We took a sample of 5 females from each group, the Shellfish Diet and the Vespa, and we chose the 2 best and the 2 worst. In the top picture, we have a Vespa, the first 2 are the best, the last 2 are the worst. In the bottom, we have Shellfish Diet, the first 2 are the best, and the last 2 are the worst. We counted their absolute egg number and also weighed their meat weight. You can clearly see that the Vespa had a higher average when it came to the absolute egg number and the meat weight than the Shellfish Diet. Around a 43 and a 64 and a 14. That's what I know you guys can read, just want to point it out. So this is our second stage, spawn and larvae culture. This process was very lengthy. You can see Kyle Edwards in the first picture chucking the oysters. We do that by hand. There's no evidence of me doing that because I cannot chuck those oysters. I can't open them. The second picture is our sampling. So here we took a small sample from each oyster, and then we later did a sex identification to figure out whether it was male or female. This picture is a picture of a male sperm. Under the microscope, they look like very, very small dots, and they're very, very active, and they move around very, very fast. The second picture right here is a female eggs. Good eggs look like small circles. Average are like they look like little mini avocados. The last picture is the strip spawning process. So we would take, after doing the identification, we take the oyster and then take a scalpel and then scrape out all the eggs or sperm depending on the sex, and then put it in a beaker to further dilute it into water to further count them individually. So this is our tank setup. The first 3 tanks we have are the Vespa, the second 3 are the Shellfish Diet, and the last 3 are the Pearl. We did 3 tanks for each group to further the results of our larvae. And this is the larvae at the 4th checkup. This is called the D stage because they look like little bees. This is our larvae growth and survival data. So for the larvae growth, we have the Pearl, Shellfish Diet, and the Vespa. You can see on the 7th day that the Vespa sadly crashed, but the Vespa and the Shellfish Diet had a steady incline of growth. On the larvae survival, we have, again, the Pearl crashed on the 7th day, and then they all were increasing in survival until they didn't really crash. These crashed again on the 7th day, but they were doing better than the Pearl. This is stage 3, post-metamorphosis and larvae culture. So this our downloader system. John built all these because he is our magic man. He builds everything for us. The downloader system is a contraption built to keep for the larvae at metamorphosis stage so they don't go out in the tanks when they're trying to settle. So here we have the airway tube. This is used to circulate the water. So when we have the algae in the tanks, that the algae is brought to the oysters' mouths as they're settling. These are the eyed larvae before metamorphosis and the eyed larvae after they settle in the microtrophs. Because our larvae crashed, we did 3 tests and they all crashed, unfortunately. So we ended up purchasing 400,000 eyed larvae from Horn Point Laboratory instead. And then we started this project on July 2nd. This is me. I look great. This is July 20th. We did count on the oysters. This is called a cone. Just figured that out. It's called a cone. And we use these to count the volume of the oysters for each silos. The silos are the round containers you saw in the previous picture holding the oysters. This is about 18 data growth. We took individual samples from each silo to further measure them and then microscope and count them to figure out how many were each in each silo. You can see here that the Vespa had much more growth in the Shellfish Diet and the pearl algae. When you put them on your finger, they're like little small dots. They're actually very, very cute. And I think this is my favorite stage because like you can see the shell development and the organ cells at the oysters. So this is the stage 3 development for their data. The growth is, you can see that the Vespa has significant more growth in the Shellfish Diet and the pearl. Survival is, it may look like the pearl had more survival, but because of the one silo had much more survival than the rest, it brought up the average. So the average between all of them is kind of at one line, but the Vespa has more in a way. Conclusion. So stage 1, our Vespa Brewster Condition faster than the Shellfish Diet oysters. If you remember from the first picture I showed you, the Vespas was more plump than the Shellfish Diet and they looked more ready to spawn. Stage 2, the results were inconclusive. We had an expected larvae crash, so this stage will be repeated next year. We don't really know why the larvae crashed. It could be a number of reasons as to why it happened. So we will be doing that next year. Stage 3, the Vespa fed larvae was significant faster growth in the Shellfish Diet and the pearl. As you can remember, the silos, the Vespa silo, the oysters were much more growth than the rest of the Shellfish Diet and pearl. So that is the conclusion. Acknowledgements. I like to say thank you to Maryland International Partnerships with Mark Street Venture Vespa for sponsoring the study, Dr. Ming Lu for being my advisor, Brittany with Brian, Kai Wood, Kyle Edwards, Miss Dr. K, I didn't want to butcher her name, so we're going to call her Dr. K, and John Farrington. Thank you so much to the audience for paying attention and giving me your undivided attention. And if they have any questions, I will take them now. Do you have a question, really?
I do, yes. So I believe one of your first slides in terms of gonad development, you were looking at just 2 of the feeds versus 3. Is there a reason you weren't looking at the 3rd?
So when we first started the project, we were only doing the shellfish and the Vespa because we were still preparing the pearl algae. We just wanted to see at first whether the Shellfish Diet or the Vespa would faster their gonad development before we started the project on the pearl algae.
Okay.
Are there any theories on the crash?
So there are many. There's nothing conclusive on why. It could be the water quality, things getting into the water. It could be spawning too late or too early. It could be many factors. We just don't know exactly which one it is.
When you said you adjusted according to the weather, the pictures look like everything was inside.
Oh yeah, they were, but we got our water filter from outside. So the water was filtered through a pump inside. We weren't adjusting them based on the weather. We were just taking notes on the weather. So if there was more storms, the salinity might be higher because, yeah, that's right. The salinity might be a different temperature. The salinity might be a different level because more fresh water is entering into the water. If we have hot days, the fresh water is leaving. So we just took notes on the different things going on outside, depending on the salinity levels inside.
Someone on Zoom has a question. No?
Yeah, I don't know if anyone's monitoring the raised hands on the Zoom, but if I may,
Yes, you may.
Thank you. I wanted to ask you about the different types of diets that you were using. Are these, do you know what species of algae you were feeding them? I imagine some of this is proprietary, so the companies may not be as open with what it is, but I'm curious as to whether you were feeding your oyster different species of algae or the same alga grown different ways.
So our amazing Brittany is not here today, but she did show me a few of the names. I don't remember the full name, but I remember like glimpses of each abbreviation. So we had like TET and mono and chagra. They're just abbreviations for the longer names of the algae. Each algae is used for different stages of the larvae. So one algae was used for them to grow their shells. Another algae was used for like the beginning of their feedings. It was small enough for the baby larvae to feed. So each algae was used for different things during the process.
So these Vespa and the other diets that you buy commercially are mixtures, or are they single things?
So the Vespa, I remember when we were feeding, we fed different things from a Vespa, different algae, but the company is a Vespa. So we'd feed them TET and CM, but we would label it a Vespa.
Okay, thank you.
Yeah, I can talk a little bit more about this. This question is too hard to for sure. Yeah, we use Avaspa in separate. So the currently main three species algae we use TSO, chagro, catodrosis, and tetracenamines. So our produce algae also focus on these three species, and the Avaspa algae we also use three of these species separately. But for the Shellfish Diet, it's a mixture, so we cannot determine that as is. Yeah, and we keep the same density every feed because all the three products, Shellfish Diet, Avaspa algae, and Cultured Algae, it did the same algae number per meals.
Thank you.
Yeah, thank you.
We'll talk more about this in January, Ming.
So just one final note on this. This Maryland Industrial Partnerships grant is supported by a special fund through the Maryland Department of Natural Resources. So I want to credit Maryland DNR for being a big supporter of this effort through the Maryland Industrial Partnerships.